Okay there, campers. Today we're going to take a look at how to do a monohybrid cross. We've been studying monohybrid crosses in class, and this would be a great way for you guys to review the steps on how to do a cross in preparation for dihybrids, which we'll take a look at next, as well as just general practice on some of your basic genetic crosses. In this case, we'll look at a monohybrid cross with the inheritance pattern of dominance and recessiveness. Later on this unit, we'll get to other inheritance patterns in the ways that genes can be expressed, so that it's not always dominance or recessiveness. In some cases, you might find other ways that genes can be expressed in the phenotype, but we'll get to that. Regardless, you're going to do a cross the same way every time. So we'll take a look at using some of the traits that we've been using in class. Why don't we take a look at ones where we had two plants that are heterozygous for green pods. From this, we'll determine the genotypic and phenotypic ratio for the offspring. So, to do this cross correctly, we're going to take you through all of the steps. The first step would be to determine the alleles you're going to use. For this one, we have to determine which allele is the dominant and which allele is the recessive. So, in this case, I know in the problem, you're not told what the recessive one is, so we'll let you know that yellow is the recessive. So for this, we're going to take a big G, since that's the dominant allele, and that's going to code for green pods. A little g will code for yellow pods, and that's the recessive allele. In dominance and recessiveness, you want to make sure that you're using the same letter, but you're going to use a capital and a lowercase to distinguish dominance from the recessive. Okay, so there you have it. Step one, all set. Step two is to determine the parent genotypes from the problem. So you're told up above that you have two plants heterozygous for green pods, and we're going to cross those two. I guess we should say that. We're going to cross. Okay, two plants that are heterozygous. Since heterozygous means that each allele is different in that particular gene pair, we're going to have one of each. So your first parent is going to be big G, little g. The other parent, again, will be big G, little g. As far as a monohybrid cross, this is about as complicated as it gets. Once you have your parents established, the third step is to determine the different alleles the different gametes that each parent, and different, that's really important, gametes that each parent will contribute, or I should say, could contribute. Because really what we're looking at here is what are the possibilities of one gamete fertilizing with another, and looking at the possible outcome for their offspring. So once again, we're going to have the parents. We have a big G and a little g. and a big G and a little g. Now remember that each of these letters are found on a homologous pair of chromosomes, and in meiosis, that pair is going to separate. So when that pair separates, you're going to find yourself with a gamete that has a big G in it. The other one, you'll find a gamete with a little g. For the other parent, the same thing applies. When that pair separates in meiosis, you get a gamete with a big G and a gamete with a little g. From this, you will take these pieces to build your square, which leads us into step 4. So you're going to build your Punnett square from the gametes in step 3. So I take those gametes and I place one parent across the top. I take the other parent and place them down the side. And I build my square. With this, I'm going to figure out what the possible outcome would be for an offspring given a particular gamete fertilizing with the gamete of the other parent. So I'll go ahead and build the square and actually determine the outcome. This is step 5. Do the square. Once the square is complete, I'm going to look at the results and I'm going to answer the problem. So step 6 would be to answer the problem given, and in this case it was to figure out the genotype and phenotype ratio of the offspring. So I'm going to just rewrite the Punnett square here now that I'm on a new page so we can see this all together. So as I take a look at this, the genotypic ratio is going to look at what are the actual genetic possibilities for the offspring. That's what the genotype is. So when I look at the square above, I have 3 possible genotypes out of the 4 possible outcomes. So for the first one, I could have a homozygous dominant. So I look and I have 25% big G, or homozygous meaning the same, dominant genotype. I could get 50% of heterozygous genotype, or I could have a 25% chance of having a homozygous recessive genotype. And there you have that. For the phenotypic ratio, I have to look at what the offspring are going to potentially look like. I could have plants that have green pods, I could have plants that have yellow pods. And when I look at my 4 possible outcomes, it turns out that 75% will have green pods to 25% yellow pods. As you look above, as long as there is one big G present in the genotype, they're going to show the dominant phenotype, which is green. The only way they're going to show the recessive phenotype is if their genotype is homozygous for that recessive allele. So there you have it, a monohybrid cross completed using dominance and recessive. Next time we'll take a look at how you do a dihybrid cross using these same steps. Hope this helps, and if you still have any questions, don't hesitate to go ask your teachers for help. Take it easy, guys.